Good morning to you all. Today I'm going to present my study on exploring water absorbance spectral patterns of biofunctional extracts in product development. This study was conducted in Unisato Aquaphotomics Laboratory, Wakayama, Japan. And here you can see some pictures of Unisato hot spring, natural hot spring. And apart from my presentation, I warmly invite you all to visit this beautiful place. And here you can see some pictures of Unisato Aquaphotomics Laboratory. It's a very beautiful place, equipped with modern equipments and a talented staff. And in this study, I'm going to explore unique water absorption patterns of natural products and their extracts using NIR spectroscopy with the focus on their biofunctional characteristics. Our main objective was, there was mainly 3 objectives: to analyze and attempt to classify natural producers using the first water tone, and second one, to assess if key bioactive properties of the natural producers are preserved during the extraction process to enhance food and cosmetic applications. And finally, to develop products with desired functionalities. In our studies, we have measured several natural producers, but here in this study, I'm going to present the results of 20 natural producers and their biofunctional extracts. And we used a micronear instrument to measure our samples with 3 repeats and 5 consecutives. And we measured basically 2 types of products. Raw products, we measured the surface and cross-sectional measurements. And in biofunctional extracts, we measured the cuvette cell, that means the extracts. And first, we did some classifications, PCA and SIMCA, to categorize or classify our studied samples. Here you can see the PCA scores and PCA loadings. And then here in this picture, you can see the SIMCA discrimination results. And here you can see a list of products we have checked. We checked some citruses, different types of tomatoes, green beans, 7 types of edible leaves, some yams and mushrooms. And in this graph, you can see average raw spectra of natural products. For your easiness, I have divided this average raw spectra into different categories based on their phytomorphological features and the classification used SIMCA and PCA. And not only that, we have measured different points of the same product also. As an example, I am showing here the result. Sorry. Okay. Thank you. And here you can see Macomo leaf experiment result. And here in the Macomo plant, we have measured 3 points, top, bottom, and the middle, to see whether to see the differences in water absorption. And we observe the least absorption of water bands from the driest part, which is the leaf top, and the most absorption bands from the bottom, which is the wettest part of the plant. And you can see the average difference spectra analysis result and echogram for these 3 kinds of parts in the same plant. And then we extracted the plant biofunctional water using this vacuum dryer installed in our laboratory. And it is operated in between 38 to 42 degrees of Celsius. And here you can see average raw spectra of different categories. However, you may not be able to see the differences in different categories. Therefore, we have conducted a different spectra analysis by subtracting purified water from the averaged sample data. And in this, by this analysis, we could found that biofunctional water characterized with less free water molecules and small water clusters, and more water solution shells, strongly bounded water molecules and water ions. And if we see the result of yams and mushrooms, it is with more hydrogen bonding of water molecules. Then we conducted PC and echogram analysis for different categories. Here you can see the analysis results for fruits data. Raw fruits and the leaves, which I'm going to show next, exhibit greater spectral variability compared to their extracts, and less free water molecules and small water clusters in biofunctional water. And here you can see the results of leaves data. And as you can see here, like in the fruits, raw leaves show greater spectral diversity compared to their biofunctional waters. And this is the result for yams. We have explored the NIR spectroscopy of carrot, beet, and onions. Here you can see the result. Yeah, they are with small water clusters and indicative of metabolic activity when compared to other categories. And this is for mushrooms. And there we have used shiitake and 2 types of kikurage mushrooms. And these mushrooms, biofunctional waters, showed more hydrogen bonding with water molecules. And after that, you can see here a summary of echograms for biofunctional water and their raw product for different categories. Red color shows the biofunctional water and black color shows the raw product. And as you can see in almost all the categories, partial functionalities and properties were preserved even through the extracts. And biofunctional water extracts showed reduced free water molecules and small water clusters, but an increase in solvation cells and strongly bound water molecules, as I have mentioned you earlier also. And here is a summary of our main study, and you can see raw products and biofunctional water in separate aquagrams. And you can see the extracts with characteristics for their category balance between hydrogen bonding, water solution cells, and trapped and free water molecules and water ions. And then I'm going to show you an example for successful product development. And here you can see the development of yogurt rose water blend. In this exploration, we use Suku and 100% rose water to create rose blend. And then by combining this rose blend with yogurt biofunctional water, we created yogurt rose blend. And you can see the result with the aquagram. The black color shows purified water, and blue Suku, and orange color is showing the yogurt biofunctional water, which is having many confined and free water molecules and less hydrogen bonding. And you can see the result in red color, which is yogurt rose blend with less free water molecules, more strongly bounded water with absorption peak at 1464 nanometers, which is in protein synthesis band. And these are the conclusions of this pilot study. Distinct water absorption spectral patterns in plant reflect unique water binding properties depending on factors such as phytomorphology, regulation of water, gas, and nutrient mechanisms inside the produce and various adaptive mechanisms. And preservation of bioactive properties in extracts from the raw produce enables the development of targeted functional products. And aquaphytomics successfully applied for a quick, non-invasive way of monitoring, exploring, and evaluating functionalities in natural systems. And in the future and ongoing research, we are exploring more natural products in order to develop food and cosmetics with the desired functionalities without losing the original quality of the natural produce. And this is the end of my presentation. I like to invite you all to visit my poster exhibited outside the venue and give your comments and suggestions for our research. Thank you very much.